this lecture, we're going to talk about diffusion models and specifically diffusion models for protein design. So let's get started. First, I wanted to show you some examples more intuitively, so non-protein examples. And diffusion models have been developed initially for image generation task. So let's take a look at an example where I can give a prompt as the input and produce some images using diffusion models. And so for this prompt, I had picked back when we were doing this in June. I just wanted to see what would happen if my input was an abstract painting of a room full of proteins learning machine learning. This is what I was able to generate with DALL-E. So DALL-E is a prompt-based image generation model, and it's also capable of sort of refining images if you have an input image. Also, you can just do some free or unconditional generation without this type of prompt. And so any observations on these generated images? Does everything look great? Okay, so these look too human. So it's not doing what we asked. What else? Some blur. Maybe the fact that I asked for a rather abstract drawing. We could explain it. What about the text looks a little off? This one's definitely cursive. This looks a little cursive. Okay, so it doesn't actually spell out a real word in English, right? Maybe it's using what I said here, but it doesn't actually look like text that we could read. So it turns out, funnily enough, these image generator models have some difficulty spelling out text in images. And so this is something that's, for example, really easy for humans, right? From the age you learn how to write, you could make a drawing with some text in it, and you would be able to spell it if you're able to spell it outside of an image. But for these types of models, there's quite a bit of difficulty getting these words to look exactly right. Overall, would you say that this prompt is fulfilled by one of these 4 images at least? Raise your hand if you think it did a good job. Okay, so let's move on to a different example. So this one was inspired based on my personal passions. And so I wanted to see, of course, I wanted to make it funny, but I wanted to see a humanized Red Bull drinking a Red Bull with a Red Bull themed fridge full of Red Bull in the background. Okay. So, okay, this is, just prepare yourself for what's about to happen. So with this, first of all, we can sort of see the same text pattern. It doesn't exactly look like the words red and bull necessarily. Maybe sometimes it gets a little bit closer to spelling it right, though I'm not sure that's an E. And then there are some other problems with this. So can anybody point out some problems with these images? Okay, so maybe the can colors are wrong. I promise this is not an advertisement by any means. So in here, I think the idea was that this this being is drinking the Red Bull, but it's too close to the face, right? It doesn't quite look like drinking. What else? Any other noticeable problems? Yeah, so the logo is not necessarily one with the can, so that's a little interesting. Raise your hand if you think this prompt is fully satisfied by these images. Okay. Okay, so some belief in the third image here. Yeah, but maybe a little bit less so than how much faith you had in the previous slide. So last time I showed this, one of the interns actually drew what it would look like if it was more accurate. And so in here you have a fridge full of Red Bulls. Okay, so Red Bull themed fridge full of Red Bull. This is in the background. And then you have a humanized Red Bull because it has presumably human hands and wearing a t-shirt, and then drinking a Red Bull a lot more noticeably and seems just more fit to this task. So would you say that this image drawn on a whiteboard by a student is more accurate to what we asked for? And so there is still some room for improvement with these models, right? What they come up with is very interesting, but sometimes the images are a little bit cursed. But also, let's talk a little bit more about how exactly do you do these things, because we can see this being useful. It's useful for wanting to refine an image that, let's say you took a picture and you wanted to make some slight refinement, maybe you want to make it look more abstract. You could have that prompt and, get to create some digital art. So this is most certainly interesting. So how do we do this? How are diffusion models trained? And right now, we're still in the image generation space, so let's talk about images. And over here, if you just focus on this figure right here, we're going to have this image of a cat. So this is our data. And then we're going to gradually add some noise to this image over time until it gets to basically fully static noise. And this process is the forward diffusion process. So taking real data, whether that belongs to an image or otherwise, and then getting to a full noise sort of ending point. And then after we do this process, we want our model to learn how to go all the way from noise to real data. So that is going to be the generation step. And it's reversing this noised process. So this is denoising, starting from static back to data. And so diffusion models will do this iteratively. First, you add noise to the set of data, and then the learning task. So what's difficult is, well, how do you step back into the right direction and make this look like a sharp image, right? So you've gone through all of these steps, and then there's going to be some time step associated with this noising process. But how can you sort of go back and make it look like something that belongs in the data set? And so the noise that you see here is Gaussian, and we see Gaussian noise used a lot. There's other types of noise that you can add to your data, but for a moment, let's just entertain this. And once we train a diffusion model fully, we can endlessly generate essentially new data that would belong in this category over here, that is hopefully high quality, just from input or randomly sampled noise. So we're just starting from noise and then figuring out other types of images. Maybe our whole training data set was based on animal images. And so just starting from noise, it would learn how to do this for animals that don't exist and don't actually have a picture. And so the task and the model is generative. And so we're generating things from essentially scratch, but based off of learned distributions. So it will still reflect this denoising process learned on your overall data set. So if your data set heavily relies on, let's say, images of creatures rather than inanimate objects, then you're going to see that reflected in your final generation step, no matter how random and whatever input you end up sampling. And so diffusion models, or more formally referred to as denoising diffusion probabilistic models, have been around for a while. However, they have become more useful and possible to do things with recently. And so that was an advance, specifically in computer science. And so this work done by Peter Abeel and others at Berkeley sort of made it more usable. And so I'm just going to show a figure from that paper where they once again go from your data and all the way, your data all the way to a learned noising process. And then in this denoising time step, you're approaching an image that looks sharp. And so this sort of directed graph is going to help your model get from noise in generation to real or convincing data. And so they train it based on lossy compressions of images. And then what they can do is, well, from this lossy compression, you fill in the blank, right? So maybe some of the pixels are missing, some things are blurry. It's a little bit lossy. You compress the image and then make it look nice again. So make it go from this to a sharper image. That is going to be generation sort of recapitulating some of the loss patterns. And so your loss function is based off of what you were able to fill out. And then you can also produce high quality reconstruction. So if you have an image in mind that you just want to sort of slightly refine and make sharper, maybe you took an image on worse quality camera and you want to make it sharper, you would be able to do that if your model trained on that kind of lower quality input and then producing high quality reconstruction of those images. And so as long as it matches the task that was asked during training time, you can do this reconstruction or recapitulation type of work. But because this is a generative model and you're sampling sort of different types of input, different types of random input noise, what's going to happen is, do you think your final produced image itself is reproducible? Can you go through the model 100 times and then always end up at this image, starting from different types of noise? So while there will be some bias towards what this final outcome is from models' learned distributions, because it is generative, it's going to not necessarily recapitulate the same type of pattern every single time. And so you will see this more so in the practice, especially when you compare to one another. Okay, so let's also take a look at what this looks like from more of a figure perspective. So we want to produce high quality reconstruction given images, and then some of these have a prompt to go hand in hand. And then you can see that our sort of final generated or produced image is quite high quality, right? Raise your hand if you're convinced by these results. Okay, so there is an extensive human sort of, I guess, professional image-based training set that is somewhat standardized in computer science. And so people tend to test with this. And back in 2018, the task may have been, well, based on this input image, can you take off the sunglasses? And you may have seen some of that. And people used to sort of get to a very high accuracy with that type of task. And so this is entirely different, right? Because we're changing what the human looks like completely. But the picture still looks like a real person, right? So let's talk about what we learned from this and how can we apply it to proteins, because we're interested in proteins. And this looks very, very cool. So if we could generate proteins based off of some type of diffusion model, well, maybe we can also get to very high accuracy, either transition, so maybe we partially add some detail to the protein backbone that we didn't have there before, or we go from unconditional free generation using diffusion models and make brand new proteins. Okay, so before we get into this, diffusion models are relatively speaking extremely new. The first one from Namrata Anand and others came out, I want to say, last spring. And so they've sort of been around since then. And this was the first application where somebody actually implemented diffusion models on protein data and evaluated in terms of protein accuracy metrics. And then since then, there have been other sort of papers. And as we speak, so for example, RF diffusion from the Baker lab here has been published 3 days ago in Nature. And also, as we speak, there is a preprint missing from this. So from Posu Wang's lab, there is another all-atom diffusion model, and that probably has been out for maybe a month, maybe a little over a month. And so there's so much current work in this space and using diffusion. And we're going to talk about these 3 specific networks and how they differ from each other. And eventually, we're going to do a demonstration on RF diffusion. Okay, so the first paper, titled Protein Structure and Sequence Generation with Equivariant Denoising Diffusion Probabilistic Models, let's get started. So in this model, what they do is they generate both the protein structure and the sequence based on a diffusion process. And so in the training, let's just skip this part for a little bit. In the training, and also if we just pay attention to the layers here, so just like AlphaFold, they have a structure module that is IPA based. And so starting from some noised structure, so some protein structure, you go through this structure module and then try to predict basically a very well organized, actually ordered backbone. And so your denoising process is to go from slightly random noise added coordinates to real coordinates. And so this is what it looks like for structure. As for sequence, what happens is they have two other things on top of this. So they place rotamers in a similar way, and they predict the sequence based on masking. So the model doesn't always know what sequence identity would be at each of these positions, and so it has to predict a sequence, and then there's a loss associated with both sequence and then structure based off of how well this was denoised going through here. And then once again, we see sampling. So with sampling, initially you have a lot of chaos here, and then it starts looking more and more like a protein, and then hopefully you end up somewhere where it has ideal bond lengths and angles and maybe fits into some torsion space. So with this model, one thing to mention is that you can give it constraints to make it more accurate. So these constraints, or the adjacency block here, is a way for the user to define secondary structure and also some contact so that the model has something to go off of. So this conditions the model. And with that, it's also important to note that the length is defined, as well as this to make it a little bit more realistic. And so if we take a look at these, so some of these samples, I mean, these look very interesting, right? Maybe these look like some of the proteins that you've looked at. And then how does it compare to maybe some hallucination examples? So if you just look at, the overall physics of it, what do you think of the structures when compared to some of the hallucinated structures? Okay, so there are some beta sheets. Okay, let's move on. So let's talk about another paper that has been out since December. Gavor Grigorian has his own lab, but this is from the company Generate Biomedicines, and from this team with John Ingram as the first author. So this model is called Chroma, and their paper is titled Illuminating Protein Space with a Programmable Generative Model. And so let's take a look at how they have this diffusion process lined up for proteins. So in this case, let's start over here. So we have a real protein backbone. This is our structure. What we're going to do is we're going to go through noising of this. So this is going to be our training sort of example, is we're going to take a real protein backbone and have some polymer collapse. So this direction is going to be the collapsed polymer system. And based on that, we want to go through and reverse this process and denoise it so that we end up with a protein complex that looks realistic. And so what is one of the differences between this versus from Namrata's paper is that you're not collapsing or adding noise. You're not adding noise at the level of atoms, but you're collapsing parts of the backbone structure to sort of the center, and then trying to figure out exactly where should the model place every single one of those subunits, let's say. And so something that's interesting with this also is they have a specific design network. So this is sidechain agnostic, and once they come up with the protein backbone through the generation process, it goes under or into a design network that comes up with the exact residue identities at each position. And so you can think of this as basically very similar to what protein MPNN does in terms of coming up with a sequence given a backbone. And so this is the purpose of this design network right here. And so in terms of noising their structure, or what to do, or how to process information from this noise structure, so the other day we had talked about having orientations, or these basically distances, frame orientations, and then we wanted to figure out sort of different edge features and construct a whole graph. And so Chroma is entirely based on GNNs, graph neural networks. We talked about protein graphs. And in here, they also have their own confidence scores associated with placing structure. And so the network learns to have some confidence with this process for the structure, which allows them to predict more realistic structures. And then, as for their conditioning, what they do is actually really neat. They have a very sophisticated way of conditioning exactly what should be generated. And you can have free generation, just like before. So when you had free hallucination, what did you come up with? Did you make changes to the structure? Did you start with no structure at all and make a structure? And then did you have to figure out the sequence? Right? So it was entirely unconditioned, and then you had to hallucinate everything. Okay, so in this case, you can do the same exact thing where you're doing free diffusion, so there is no constraint, right? There is no input backbone necessarily. So what you're starting with is basically nothing, and you want to find a protein shape. So give me a protein geometry that sort of reflects what you've seen from your training process. Okay. But what if we wanted to condition that generative process? What if we wanted to have some symmetry? So what are the types of symmetry we talked about in regards to proteins? Dihedral, cyclical? Okay, so symmetry, maybe we're interested in defining a symmetry, and that can be our constraint. So we want to figure out how to diffuse a protein that has, let's say, D4 symmetry, right? And then you could also have some substructure. So what do you think this means? What's a substructure in protein structures? Okay, so you could have some part of the protein already known, right? What's a relevant design case for this? When do you know what substructure to keep? Okay, so maybe you would want to keep everything but the loops, and so you end up designing the loops, makes sense. And then shape in general. So can you have other types of constraints defining a shape, a general protein shape? So maybe something about radius of gyration could be incorporated there. That makes sense. And then they add something called semantics. So we're going to see that in a second. Okay, so these are some proteins that were generated with Chroma. And so I just wanted to show you some of these sampled examples. So yeah, how about you just maybe take 1 minute to discuss? Any one of these protein structures or protein complexes stand out? Okay, this reminds you of collagen. Maybe a little bit of coiled in here. Okay. Are there any symmetrical proteins here? Do you see any symmetry? Do you see any monomeric proteins? I know you can't see the sequence, but and we also can't really, I guess, see which one of them are really, really close. But is it possible that with this process, if you can define some symmetry, you could also have heteromeric assemblies? So maybe you have homomers with some symmetry, and then you could figure out how to bind something. So what if the partial input from the homomeric unit is actually an interface with another protein? So you could add on, and then since it works so well on these types of complexes, well, maybe we can create complexes that have both homomeric and heteromeric sort of protein structures in the assembly. Okay, so let's talk about some of their results here. So using Chroma, they generate structures. Using the design network from Chroma, they design a protein sequence onto this given structure. What do we call this problem? What is this process? So this is fixed backbone design, and then I take a protein backbone with a design sequence and then go here to predict the structure. What is this over here called? So this is structure prediction. And then what if I take my generated structure and compare it to the predicted structure? Well, maybe that tells me something about how well my model can generate structures such that they can be predicted with an independent structure prediction method, right? It also indirectly evaluates your design process, because if the sequence is problematic, designed onto this nice generated structure, then that's going to mess up your structure prediction. And also, once again, indirectly, it evaluates your structure predictor, because while we have highly accurate structure prediction methods, there are still some cases that we observe they can't quite get the right structure, right? So what if we generate a very novel protein structure using Chroma and then design the correct sequence, but can't really get any structure prediction method out there to get the correct 3D form, right? Could that happen? And so this, to me at least, sort of signifies the importance of evaluating experimentally, because you could determine where you're introducing that type of error. But overall, it's definitely a good sign to have all of your structures predicted and be very close to the generated structure, because then all of these things are working well in tandem, right? And so to do this, they're over here showing that they've taken a TM score. Okay, so we know how close the structures are to each other, right? Based on the structural alignment. And then the one they report is 0.8. Okay, let's move on to panel B. So in here, they're showing, based on Chroma generating proteins, so some of their generated proteins, protein length compared to the prediction TM score. And so they're able to show and sample a bunch of different lengths. And then we see that there is some that do quite well when compared to the structure, when compared to the predicted structure. And then there are some that don't really align as well and have lower than 0.5 TM score. If they're lower than 0.5, do you think that they're the same structure? What is our cutoff? Or what's a reasonable cutoff for TM score? And then you have two structures that start looking too different, and we no longer would consider them to be very similar. Well, what do you think about 0.5? That's low. So these, you would say, they're entirely different backbones, right? Okay. So at this point, do we know whether Chroma just generated proteins that wouldn't fold at all? Or do we think that this sort of difference or discrepancy between the predicted structure and the generated structure is sort of introduced by the structure prediction step? We don't know, right? We wouldn't know the answer to that. We just know that for most of them, there is some high agreement, and we can sample different lengths. Okay, so let's move on to panel C. In here, they're just showing the TM score of predicted structure, and then frequency in terms of some model parameter here. And then overall, you can define cutoffs for what parameters you want to use, and what TM score or other type of structural alignment metric is the most relevant. And then over here, they're just showing or highlighting some generated and predicted structures, as well as their TM scores. And then you can sort of go through these and see if you notice anything sort of interesting about that. I want to talk about something called terms. So terms also have been around for, so these have been around for quite a while, and terms stand for tertiary motifs. So they're functional groups in proteins that you can split up and sort of build most of the PDB from. So most native proteins can be built by these individual tertiary motifs. And based on these functional groups, you can sort of make up, or based on these groups, you can make up most of the backbones. And so if we study the term decomposition of chroma-generated proteins and compare it to the native, then we'll be able to tell whether they can sort of recapitulate these terms or tiny motifs we normally would see. And maybe that's either sort of step in the right direction in that we're still remaining in that realistic protein backbone space, but also if we have the capacity to diversify what possible motifs we end up designing and they're not seen in nature or already in the PDB, then it's interesting that we have the ability to go that far. And so in here, let's just talk about one of these. So they're showing what happens when you compare the native sort of RMST to the closest term versus the Chroma models. And you can see that it's able to cover or span a similar range to the native while also having the capacity to sort of step away from it, so become more distant from the closest possible term. And so this is interesting and also very useful if you wanted to optimize and figure out, well, at what point can you remain diverse but also experimentally successful? And so let's take a look at some of the symmetry that has been generated with Chroma. So these symmetric assemblies, first of all, they look really pretty. And they also tell you what kind of symmetry it is over here. So if you need a refresher, I probably can't say all of these names, but you get the point. And then, of course, this one is gigantic. So is there any use to making these types of assemblies, these symmetric assemblies? Is there any biological purpose to these? Cages, okay, yeah. All right, and then in here, first I want to show you how they use substructure. So this could be a motif, so you did an exercise on this. It could also be a part of the protein, so you've actually given antibodies an example, and they also talk about that. So your idea would be to keep a part of the protein constant and then sort of generate around it. And so they demonstrate that they can do this. So let's say you want to keep this alpha beta beta motif, then you can build a protein around it, but keeping this motif as constant as possible. And so this allows us to have a lot of control over, especially in terms of binder design or protein scaffold design. And then for this shape, what they do is very interesting. So they actually just diffuse proteins based on some conditioning on either so this is just the alphabet, and these are some numbers. And I think it shows that we have great control, even constraining from shape that we wouldn't necessarily see in protein examples at all. We can still come up with these very interesting overall assemblies. And so do you see anything in here that looks like it could actually exist? It's a little bit difficult, right? Maybe some of the packing or some of the hydrogen bonding isn't necessarily satisfied in each of these scenarios. And so, yeah. Can't that lie exist? This? So it could. The problem with it would probably be the lack of packing at that helix, probably, but something. And then do you think most of these would pass radius of gyration in the way we looked at some of the hallucinated proteins? Depends on how much you allow it, right? But yeah, it's some interesting work and definitely worth looking more at the paper. And then I'll also talk about the way they construct the protein graph is a little bit different. So the neighborhood and the way that they select the neighboring residues or the nodes is different. And so this is what a KNN graph looks like. So this would be your neighborhood given some size, some possible closest n neighbors would look like this. And then in a uniform graph, starting at this point of origination, you would uniformly pick amongst all of the possible nodes. In exponential, it would look something like this. And then you have inverse cubic. And so what they did was they optimized on this neighborhood-based graph so that they could make their graph more informative to both structural and design related loss. So starting from this, you have some k and n, so 20 in terms of starting nodes here, and then your closest, but then 40 that are inverse cubic, so some inspiration from this and some from that, and it turns out this mixed graph and what they call graph topology ends up being sort of the most accurate way to approach this problem in this architecture. And this is what the architecture looks like. But right before that, let's also go over this portion. So what they have implemented, which I thought was really cool, is they have some neighbor consensus. So based on, to determine the position of I, they will rely on neighbor J and neighbor K's information consensus. So wherever there is overlap in terms of positional information for I, there will be sort of the possibility of placing something here increases because they're in agreement versus when they're not in agreement. Obviously, you could expect all sorts of problems. So it makes it more realistic in terms of placing exactly where each of the, let's say, alpha carbons should go. And then I'm not going to go into super detail of the, I'm not going to go into the backbone network or the design network, but they're based off of this GNN layer that they defined. And you can think of this, some of the updates being similar to protein MPNN, for example, especially for design. And then the nodes and edges, so you're familiar with the concept. And then if you're curious, you can look at the specific operations that take place and how everything feeds together and what updates what. But essentially using that, they have one network that's capable of producing and generating, so this generates new backbones, and they have one capable of taking in backbone coordinates and coming up with sequence as well as the rest of the atoms. And so placement of sidechain atoms plus you determine the sequence. So those all happen at this design stage. And now let's move on to the third paper that we'll be covering, which is from the Baker lab, and this is called RF diffusion. So it's RF for Rosetta fold. RF diffusion is based off of Rosetta fold, which is also from the Baker lab. So let's look at their this figure from them right here. So in their diffusion model, they're starting, once again, they're going to start from some protein structure. So this is well-defined, real protein structure, and then start adding Gaussian noise to this. And then once we go over this forward or noising process, the model is going to learn how to reverse it by placing the atoms where they should actually be and having some structural loss associated with this process until we get here. And then once it is fully trained, it's able to start from just noise and go straight to protein structures that look like they would be well-folded or well-structured. And their paper is, it's right here, and I will also share it with you. So that should be fine. And here they highlight the differences between Rosetta fold and RF diffusion. So in Rosetta fold, it was developed for structure prediction from input sequence. So they also added the capability of infilling parts of the sequence instead of everything having to be inputted. So going from input sequence all the way to predict structure, this is the task for Rosetta fold. As for RF diffusion, you have these diffuse coordinates, and based on those and no sequence information, you figure out how to make a protein structure. And then we could then take this protein structure and figure out what sequence it should have. So this whole process, once again, is sequence agnostic. And RF diffusion actually in the notebook that we're going to do a demo on as well, they use protein MPNN as the design network. So up until this point, you have a protein structure, and then you figure out what protein sequence should go with that structure. And then you could do structure prediction to further computationally evaluate all of that. And so once again, different ways you can use RF diffusion, and all of these we're going to sort of divide up once again and have everybody try out something slightly different. So you could have unconditional generation with RF diffusion. You could make symmetric oligomers. So you define the symmetry, and this is, you'll have a lot of options here. You also define how many sort of biounits go into the symmetry. Binder design, once again, you have to define the target, and then you diffuse a protein around that, making interface with the binder. So this example is for motif scaffolding. You have to design a functional, you have to input a functional motif that you want to be sort of scaffolded by the generated protein. And then this final option is a combination of this and that. So symmetric motif, so there is some symmetry to your input motif, and you want to continue and build off of that symmetry. You can also do that with RF diffusion. And in here, I'll just show you that. So in their breakdown, you're going from some noise. Let's say we're placing some of the secondary structure is coming into place here. So you can see this over here, the yellow helix take up a little bit of shape, and essentially your entire protein structure is sort of coming together. And so based on the inputs, that's super sort of disorganized and chaotic and a lot of noise here, you get to a beautiful prediction, and the structure hopefully looks like a real protein. And so why would we be interested in doing this at all, and why use it for protein design? So let's go back to the 4 cases that we talked about during hallucination. So the first one is unconditional generation. We're just interested in making new protein shapes from scratch. So we want new geometries, because if we could expand the libraries of structures that we have, then we can search for more possibilities in all sorts of applications. So that's one. Number 2 is, once again, we know our target, we know we want to bind it, maybe we would know the interface, but we want to make new proteins that are successfully binding it with high specificity and also high affinity. And then scaffold generation, once again, maybe we know exactly what motif we want to borrow from nature, so that part is clear to us. We just don't know what de novo protein can be designed around it. And then in this 4th possibility here, something really cool you can do with RF diffusion is partial generation. So you give the full structure of your protein, and then you sort of refine that structure. So you diffuse a little bit. So you add some noise and then denoise, but it still looks relatively close to your given structure. So you do some modification. You can think of it as reconstruction. So just like image reconstruction, you're reconstructing this protein. And can you think of a case in where this 4th bullet point is applicable? Why would you want to only partially redesign? So maybe you have the bulk of your structure, you're just not sure about a region, right? And you just want to see if you can generate something that looks more convincing at that region. So maybe you want to stay within a certain protein family, but sort of see what other artificially sort of generated protein looks like it would belong in that family. All right, and then you can also do this once again with monomeric, homooligomeric, heterooligomeric specifications. So you get to decide what kind of protein it should be in terms of whether it's single chain or multi-chain, and what kind of multi-chain protein. So we're going to get started with the RF diffusion demonstration. All right, so let's get started. And this is the end of the recording.